back, students. Here I am again at the whiteboard. We are in the middle of glycolysis. Remember, we're looking at how glucose, the food that we eat, is oxidized, partially oxidized, in the steps of glycolysis. This is a part of the bigger process of aerobic cellular respiration. And I think I left you at the end of step 6. At the end of step 6, we have 2 molecules of 1,3-bisphosphoglycerate. And we have 2 molecules if 1 molecule of glucose is going through aerobic cellular or going through glycolysis. By the way, how do you like this whiteboard? I posted it in my basement, so I have to kind of move out my furniture to make these videos. I don't know. We'll see if it works. If it doesn't work for us, I'll do something different in the future. All right, let's start thinking. We are on slides, I think, 16, 17, and 18 in our PowerPoint presentation if you want to add notes to those slides. And we're in glycolysis continued. Okay. We're really at step 7 in this process, and what we have after the first 6 steps of glycolysis is we started with glucose, and we now have 2 molecules. I really could put a 2 here, 2 molecules of this thing called 1,3-bisphosphoglycerate. There she is, a 3-carbon molecule with 2 phosphates attached to it. Those are going to be useful. We've made some NADH. We've made 2 molecules of NADH, that's our shuttler of high energy electrons, the tennis balls, right? And it's going to be used later to, I hope, make a bunch of ATP. But we haven't made any ATP yet, so we need to make some ATP, and steps 7 through 10 is where it's at, all right? So in step 7, 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. I better write that down. You don't have to memorize every single step in this process. Just understand the basics, right? And in this process, what I'm looking at here is the loss of a phosphate from the first carbon, right? What really happens here is there is a transfer of that phosphate from 1,3-bisphosphoglycerate to ADP. ADP, adenosine diphosphate, picks up the phosphate and is converted into, you named it, ATP. We've made some ATP here, all right? There's a transfer of a phosphate from one molecule to another. What kind of enzyme is used for transferring phosphates from one molecule to another? Kinase enzymes. That's right. This one is called phosphoglycerokinase. I'm just going to write in a kinase enzyme. So this is an exciting step because we've actually made some ATP. I'm going to write that down here as a few important things. So we've made, how many ATPs? It looks like 1 in my picture, but remember, if we're following 1 molecule of glucose through glycolysis, we've got this reaction happening twice, all of these reactions happening twice. So we make 2 ATPs. Okay, that's a great source of energy there. This, oh, by the way that we make these ATPs, there's a special name for it. It's called substrate level phosphorylation. That's the mechanism by which we are making ATP. I'm going to write that down. Substrate level phosphorylation. Okay, and what that essentially means is ATP is being made as a phosphate is being transferred from a substrate to ADP. Substrate level phosphorylation. There are other kinds or other mechanisms of making ATP. We haven't seen them yet. We'll see them later in aerobic cellular respiration. I'm going to write down that we needed a kinase enzyme to catalyze the transfer of that phosphate. And I'm going to write down that this happens 2 times. So we're making 2 ATPs here, right? And we're making 2 molecules of 3-phosphoglycerate. Okay, that's exciting. Step 8. Step 8 is a rearrangement step. What we're going to do in steps 8 and 9 is make our molecule very unstable. And in step 8, we move this phosphate from the third carbon to the second carbon of this molecule, right? So we convert 3-phosphoglycerate into 2-phosphoglycerate. Essentially, we are rearranging the molecule. That's what I'm going to write down in my notes. Rearranging the molecule. Step 9. And by the way, this does require some sort of enzyme, and so does step 9, so does step 10. They all do. Every single step. If you're taking biochemistry in your later career at Grand Valley, you're going to need to learn every one of these steps, but we're not, so let's not worry about that right now. So in step 9, think of this again as a rearrangement step to make this molecule unstable. In step 9, water is removed. So water actually leaves the molecule, and a double bond is formed between the second and the third carbons here. That double bond makes this molecule very unstable, and this molecule is called phosphoenol pyruvate. Phosphoenol pyruvate, or PEP, is the name of that molecule. And it's a very unstable molecule. What's unstable about it is essentially this phosphate is not going to stick around. This double bond between the two carbons is not going to stick around. We're setting up this molecule to dump its phosphate, if you will. Dump it to ATP, all right? So think of this as a reaction step that makes our molecule very unstable. And then step 10 is exciting again. They're all exciting, right? Okay, in step 10, we're using what's called pyruvate kinase as the enzyme, a kinase molecule again, to transfer the phosphate from PEP to ADP. ADP picks up that phosphate, becoming ATP. We've just made some more ATP. Okay. What happens is this double bond between the two carbons here is very unstable, and it quickly shifts to make a double bond between the carbon and the oxygen, the second carbon and the oxygen. And the phosphate is removed. The phosphate is transferred. What we've got here is the making of 2 molecules of ATP, because remember, this is all happening twice by substrate level phosphorylation. The transfer of a substrate from one molecule, the transfer of a phosphate from one substrate to ATP. We are using a kinase enzyme. This time it's called pyruvate kinase.